Cool. Thank you for the introduction. It's lovely to be invited to participate in this. And so I'm just going to start briefly. I'm just going to touch base a little bit on some parts of my own career path that led me to where I am today, kind of working at the interface between what you might call wet lab research, where you're sitting at a lab bench, pipetting things into and out of tubes, and also computational work at a computer to sort of understand the structure of the data. So briefly, so I started out after college. I was actually a research technician a few blocks up from here at the University of Pennsylvania School of Dental Medicine, and we studied stem cells basically derived from your neural crest, primarily bone marrow stem cells, and we sort of compared how those cells respond to different drug treatments, and we also compared basically stem cells in both your jaw bones are from the neural crest, but the stem cells, your bone marrow stem cells from your hip are not. So they have, based on sort of the ecological and developmental properties of where they originated, they have different properties when they're exposed to different drugs. So that was one of the things that we studied. Following that, I got my PhD degree at Boston University, and this was a little bit different. So I moved on from studying cells to the whole organism level, and my thesis basically researched in two sea anemones, so kind of whole organism level. They had differences in how their developmental trajectories played out. One of them, after hatching from an egg, basically turned into a swimming larva and then would settle. That would be this one over here, and another one had a parasitic stage. And so I studied how gene expression, this is visualized in a heat map here with different colors indicating kind of different levels of gene expression, how that differed between those species and how that correlated with how the sort of the manifestation of that in their life history trajectories. So this involved work in the wet lab. Here I am posing for a photo in the lab bench, and here I am. One of the ways that you collected them was just like a little sieve in the salt marsh. So kind of got to combine research in the lab and then also field collection research. Recently, since 2014, I've been working in Junyoung's lab, and so I've kind of returned to my roots as a cell biologist, and this has been a very fulfilling experience for me because I get to continue to do wet lab research and also bioinformatics research a little bit. So I'm still sort of at the nexus between these different types of modalities of research. And so this is a picture of a neuron. It's a mouse neuron. And this is the mouse neuron stained for different proteins and also different RNA molecules that we can visualize with microscopy. We do a lot of sequencing. This is a picture loading a sequencing flow cell onto the type of sequencing instrument that we have in the lab. And this is some sort of visualization, statistical visualization of different cells grouped by their gene expression profiles. That's basically sort of an overview of what my path to where I am right now. So briefly, some of the things that are going on in Junyoung's lab, and this is not an exhaustive list. This is just a small list, but it's interested in sort of comparative genomics of mammalian neurons, and also at the single cell resolution, looking at cell differentiation and also variability of mRNA expression in neurons and various other cell types. So I'm going to just do a little overview of one of the projects on which I've been working, and sort of starting out with the observation that cell phenotype variation is fundamental to the function of a multicellular organism. We all have different types of cells in our bodies, and that contributes to how our bodies function. And so these processes generating the diversity of different cell types are active throughout animal development, and then also as you get older and your general sort of maintaining your body homeostasis, you have to replace these cell types, they age, and so this is critical basically throughout the process of being alive. It is also critical throughout the process of being an animal. Regardless of what type of animal you are, animals are composed of many different cell types. Some animals have a more complex repertoire of cell types than others. The sea anemones I studied for my dissertation were relatively simple, both morphologically and with regard to their different cell types. But in general, phenotypic diversity of cells is important throughout the animal kingdom. And so, sort of extending this to the concept of an ecosystem, the different parts of an ecosystem play a role in how that ecosystem functions. And if you change those parts, if you have fewer predators or less predators, that's going to affect sort of the functional output of that ecosystem, whether it's producing different biomass or it's going to have a different assemblage of other sort of animal and plant parts to that ecosystem. And sort of extending this back to an organism level, we know that it's important at the organism level. And we know that different, wildly different types of organisms, like bacteria and vertebrates, come together to produce functional outputs. And whether two examples I chose would be the light organ in deep sea fish that produces light, and that's sort of a symbiotic relationship between very different types of animals. But that's necessary for that organ to function, which also has a functional consequence for that fish. The same thing, there's a burgeoning recognition of how our sort of bacteria that colonize our gut affect so many different aspects of our own health. So this is actually a diagram of an experiment that was done prior to me joining the lab, but it kind of illustrates the general approach that we're taking, that I'll be taking with some of the projects in the near future. And so if you think of embryonic stem cells, they differentiate into many different phenotypes of cells that are important in different areas of your body and in different organ systems. And so what they did is they sequenced, this shows how many individual cells were sequenced from different types of tissues, different types of cell populations within tissues. And basically, this statistic is a measure of the variation among the population of cells that were sequenced. So of those 19 cells for a certain adipose tissue or pyramidal neurons, how different was one cell to another, and collectively, how different are they to one another? And so if you're looking and thinking, there's a variability of, cells that, you might think are doing the same thing, neurons or heart cells are, being the cell type that they're supposed to be, they're doing the same thing, but there's perhaps a greater variability in at least the gene expression among those cells than might have been previously appreciated. So one thing that this leads to, and this is the project on which I'm kind of working for my postdoctoral research, is that variation functional? Is it entirely random, or in certain organ systems is greater or lesser variability sort of a property of that tissue that enables the tissue to function that way? And sort of extending that idea, if it is required, say for a complex system like neural tissue to have wide variety of cell gene expression states, if that's critical for neural tissue, you might imagine that it's a property that's shared among neural tissue in different types of animals. So one of the things that was done is you sequence neurons from mice and rats, and the first thing you might want to do is just look at what genes are, not even looking at variability, but what genes are common between them, what sort of gene expression patterns might define a neuron, and how similar is that between neurons of different species. So this is just Venn diagrams indicating overlap of genes that are found between neurons from different species. And one other thing that I've been working with collaborators on is sort of extending this outside of just the general rodent family into other members, more primitive members of the vertebrates, or more evolutionarily distant members of the vertebrates. We have some fish cells, and we did the same analysis and tried to sort of figure out what genes are expressed in fish neurons that are also expressed in mouse and rat neurons, to kind of come up with an understanding of what genes might be critical to being a neuron. Now we can come up with a list of genes, and by looking at the name of the gene and through using some of the databases that you guys will be going through later, you can come up with a pretty good idea for how these genes are functioning, and maybe have some hypothesis for what function those genes are imparting on those neurons that's critical to the state of being a neuron and doing what neurons do. There we go. And then one of the upcoming things that I'd like to do is sort of more densely sample the mammalian clade. So we already have, we have a reasonable number of neurons from mouse. We have about half as many from rat. And so we'd like to get more to sort of increase our sample size. If we're trying to understand particularly variability among neurons, we need to count more neurons. We can see how how different they are from one another. And we'd also like to extend it to other types of rodents. So we're in the process of writing up a protocol to do this in guinea pigs, so we can see, if neurons are highly variable as a population in mice and rats, we would expect to see that in guinea pigs. And this is, I think, an exciting thing, particularly within mammals, because mammals have such a wide range of different behavioral phenotypes. Some are very social, some are not very social, and so I think if we can sort of try to capture that diversity, that's a pretty exciting thing to be able to do from the level of understanding, how populations how populations of cells sort of contribute to the ecology of the tissue and how that affects an organism's fitness and their behavior. And that's it for my brief overview. If you have any questions about the video that we watched, I'd be happy to talk with you guys about that. And then maybe at the end of the individual, if you guys are working in pairs, at the end of that, I have all the tabs open for all the databases, and we can go through those if you have any questions on that too.
So you're saying that even within, let's say, cranial nerve number 7, all of the neurons that are running, that are part of that nerve, there's going to be great variability within those, between those?
That's a good question. I don't know. We're trying to assess, to what extent variability within even how far that goes down within the subpopulations. I don't know the answer to that. Yeah.
What's the ultimate reasoning behind your research? Is it to cure paralysis kind of thing? Is it?
Yep.
Is it?
Cool. So so I guess if you're using the example of trying to understand cellular heterogeneity and how that has sort of consequences for human health, if you think of your skin, all of your skin more or less is exposed to similar amounts of ultraviolet radiation, but you don't get freckles even, you get them in certain spots. So certain cells are more susceptible to, they respond differently to that stimulus. And that sort of same concept applies to other organ systems as well. If you get a neurodegenerative disease, Alzheimer's or Parkinson's, it's not as though you get it in every cell in your brain at once. It kind of happens in a patchwork fashion. So these cells are responding to their environment in different ways, and that affects the disease progression. And you can also kind of extend the concept to thinking about a threshold for the population of the cells, how they might respond to a chemotherapy drug. If you have higher variation versus lower variation in how those cells respond to that drug, that's going to have effects on how that patient responds to the drug treatment. That's kind of where we're going with thinking about the health consequences of that. And then even if not even in a drug system, but just as you age, certain parts, certain organs, certain systems, they age differently. So that's, I think, an interesting side shoot of that. Yeah.
Oh, in your Venn diagram, you show gene expression. Is that a specific gene expression that occur in all certain group of organisms?
Yes. So that was looking at in the mouse neurons and the rat neurons.
There were about 100.
Exactly. Yeah. Just counting the genes, which similar genes were, or which set of genes were found in both of those based on the RNA sequencing of the expression of those genes. And for that, it was actually really helpful to use the homology database, which is one of the modules. So that was actually really helpful, particularly when when we incorporated zebrafish.
Bioinformatics, do you find the gene that is expressed?
Yeah. And particularly when we added in zebrafish, because it was helpful it was kind of like a Rosetta Stone for figuring out, how do you know it's the same gene? And that was really helpful for us. Yeah, and then I'll get to you.
You mentioned briefly about the sequencing. Could you go, I'm a chemist, most of these people are biologists. Could you go through a little bit about, now have you used the sequencer yourself? Obviously, was that a picture of you?
It was not a picture of me, but I actually am the person in our lab who does the sequencing.
Can you elaborate a little bit? Do they have to be ultra clean in doing this process?
That's a great question.
Is everything sterile, or how?
Yeah,
You
so
got
I
a
can
little bit
talk.
on how this machine or
Yeah,
the
sure.
thought process on how you
Yep.
utilize this.
Yeah, I can talk a little bit about how I handle the samples and put them on the machine, and then I think there's RNA-seq module coming up.
We did RNA-seq yesterday.
Okay, cool. So this is the machine that does RNA sequencing, but it can do sequencing of like RNA samples or genomic samples as well. It basically, the flow cell is somewhere. I saw the case for it.
We saw.
Okay, cool. So yeah, so you saw the flow cell. So I don't do that. If you walk in here, everyone's like wearing like
Yeah, I was going to ask
super
about
crazy
relatively-
like coats and everything.
Yeah.
I mean, I wear a lab coat and like protective stuff, but it's not. Amazingly, it's not that sensitive. I mean, the instrument is very sensitive, but basically, the nucleotide molecules, they need a specific sequence on them to bind to it. So if there's like random like DNA or bacteria stuff falling on it in the air, it's not going to affect the sample when I'm loading it. Within reason, you want to be as cautious as you can. And the flow cell is a contained device, so the sample, I'm working on it in little tubes, and then I load it from the tube into the instrument. But once it's in the instrument, it's all done through small, thin, like capillary tubes. So all the liquid is transferred once it's in the instrument in a pretty contained environment. Does that help? And it also
You could
involves.
elaborate a little more. I mean,
Yeah.
after you've made it, then what do you do?
Fair enough. All right. So after you make the library, which contains complementary DNA, they're going to have little barcodes, and they're going to bind to specific positions on the flow cell. And it's called sequencing by synthesis. So basically, it's going to sort of flush the flow cell with different nucleotides that are going to bind to the sequences that are already bound on there. And they have little fluorophores on there, so they can be excited by a laser. There's actually, in this machine, it's a little bit different. There's only 2 lasers. In some of the other ones we've worked with, there's 4 lasers, each in a different color.
Mhm.
That helps you know which base has been incorporated into the sequence. This most recent one we have has 2 lasers, and it's like a little bit, instead of having a specific color for each base, it's like 2 bases get different colors, and then the other bases are inferred either by having shared colors show up or an absence of color. So anyway, so basically each time it's registering a color as it flows nucleotides across the flow cell, it knows, or it has a certain confidence in knowing that that's the base at that position, and it kind of just builds it up cycle by cycle. We run these for about 75 cycles, so you get to know about 75 base pairs of your sequence, what they are. And then once we have that, we send that sequence data off of the machine over to our sort of computing environment, where it's run through Bioinformatics Pipelines that the lab has developed that basically help us make sense of that.
When you said it goes through 75 cycles, how long would that take?
That takes a little less than a day. So,
That's pretty good.
with a caveat, we do 75 cycles, and then we do another 75 cycles, so you can kind of read backwards on it.
Mhm.
So the whole process takes less than a day. So if I set it up in the afternoon, by kind of the time I come in the next morning or around mid-morning, it's finished, and we'll have the data, and we can use that data for downstream analyses. So it's reasonably quick turnaround. So that's nice. Yeah, actually, I think behind you there might have been a question.
Where are we in regenerating nerve cells? Have we got anywhere? Because I have a friend who's a quadriplegic, and we do a lot of research, and there is a doctor, I forget his name, but he came out with some sort of medication that's in clinical trials now, some sort of drug. And then there's other stuff, there's this laser therapy and some other things. I mean, I see in that video we watched and stuff. I mean, it seems like you're really focusing on, like, you know, what's the difference in expression between a skin cell and a nerve cell, and how do we turn on, off, and make them similar? Has there been any progress that you could talk about? Or?
Junyoung, do you want to take that question?
This isn't our area of specialty, regenerative medicine. That we study the basic process of the nerves and the neurons, which is a somewhat different topic from what we call regenerative medicine. Right. Now, in animal systems, it has been successful in terms of doing things like stem cell transplantation to recover from, say, vertebrate damage and so on. So the human cases are much more iffy right now. And so first of all, the source of the neurons is very difficult, right? So so the idea is that if we inject somehow neuronal stem cells, so progenitor neuronal cells, that they will go in and integrate properly as well as proliferate in the right way. And the first problem is that where do you get that from, right? And so what people have been doing is doing things like using mesenchymal stem cells. These are the stem cells that you get from when you do things like bone draws, right? And so those are the standard things that people do when they do like leukemia therapy when you're trying to replenish the immune system. And whether you can take mesenchymal stem cells and turn it into neuronal stem cells by various inducing factors, and that has been very controversial. So people have done experimental therapies, especially not in this country. And there are lots of people working on this problem of converting cells of different origin into stem cells of the targeted cell type. And this is, of course, the problem that I talked about yesterday, that we start out with a single, homogeneous fertilized egg, and somehow we become these thousands of different types of cells. And that problem has to be solved before we can sort of get to the regenerative part. The reason it works in animal systems is, of course, we kill the embryos to get the proper stem cells, and we can do the transplantation. We obviously cannot do that in a human system.
I'm sorry. So you have background in dental embryo. Can you explain to me this how Cristiano Ronaldo, he usually visited, like he's been visiting a dental dentist to get regenerated from an injury he had. So he did that like last year to get back very fast.
Did he have a facial injury or just
in
general? Okay,
a general injury.
Okay, I think that's actually probably similar to what Junyoung may be talking about.
Yes.
In maybe other countries where you can get mesenchymal stem cells for therapeutic reasons.
Yeah, so he's been doing that. So he's injured right now, so that's what he's been going to like his dentist for getting regeneration from his leg injury.
Okay, interesting. Yeah, my experience with it when I was at the dental school here was the stem cells that you could get from your jawbones, these mesenchymal stem cells do have properties to differentiate into different types of cells, and we were exploring that for not necessarily a sports medicine use, although that's probably a really lucrative use for it. We were looking oftentimes for certain cancers and for osteoporosis. They give you a certain class of drugs called bisphosphonates, and they found, there's actually a New York Times article on this recently, not citing our own work, but the topic in general is that type of drug can cause cell lesions of cell death in those areas, and so we're trying to understand why maybe certain types of bone marrow stem cells might be more susceptible to that than others. That's my background on that. Should we do one more question? I don't know, how are we doing?
Yeah. Go ahead, Sean.
Yeah, earlier you were talking about the results of the sequencer, and I was kind of wondering, the three-dimensional chart that you showed earlier, is that what the machine ends up just giving you, and you have to make sense of that? Or does it analyze it by comparing it to something? I'm just kind of missing that piece there.
Yeah, no, that's good. I didn't include that intervening piece. So if you imagine like, this is like a linear progression, we get the cells, we get the cDNA libraries, we sequence them, that then comes off and you have a bunch of sort of short stretches of DNA sequence information. And that's basically what you get. And so then the next step would be figure out what those are. You know, it's really sort of, we don't know right off the sequencer what those are. So then you want to try to align them and figure out where they might match. If you're thinking of it like a puzzle or something, where do they come from in the genome? And once you do that, you know, the sequencer will produce millions and millions and millions of these quote unquote reads, these stretches of DNA. And so because you're getting so many of those, you can come up with a pretty good idea once you're sort of looking for a match in the genome of what genes were being expressed. And basically you can get sort of, you can almost count it that way. And that expression data you can then feed into various statistical algorithms to try to understand certain attributes of how your cell, if you were to project it into different dimensions, how your cells might fall along those dimensions, be similar or different to one another. So that was an intervening step that wasn't shown in the images.
As from yesterday, eventually with RNA sequence data, what we often do is after all this processing happens, the sequencing data from the sequencer turns into a set of numbers, which are the expression values for the genes, and typically we get about 40,000 different numbers for the about 40,000 different kinds of transcripts that are in the cell.
Again, I'm just like thinking about all of these numbers over and over and over again, and looking at these charts and trying to figure out, like, I'm just impressed overall.
Junjun, what would you say to kind of speak to that point in the terms of the information content that's generated now versus 2000 with the Human Genome publication?
You mean in terms of efficiency of obtaining the information?
Yeah, efficiency or just quantity for even the same.
We certainly have had more than, I think as I discussed yesterday, more than 1 million-fold increase in efficiency of being able to gather the data from the about year 2000 or so.
And that costs what, like $3 billion to do that?
Right, because you can just actually cost is a pretty good measure of the efficiency that we have. So it goes from $3 billion to about $1,000 right now. Right? So.
Yeah, that's crazy.
You want to get it?
Yeah.
10 seconds.